All right, let's get right into it. Think about every single cell in your body as this non-stop, bustling city. To get anything done, the main library, that's your DNA, sends out millions of little temporary instruction memos. We call these RNA. But here's the catch. These memos are literally designed to self-destruct almost instantly. So how in the world are scientists supposed to read these messages before they just vanish? Well, they had to figure out a way to make a permanent copy.
You know, for a very long time, this was basically the law of the land in biology, the central dogma. The idea was simple. Information flows in one direction, from the permanent DNA blueprint to that temporary RNA memo, and then finally to the proteins that do all the work. It was seen as this rigid, one-way street, a fundamental rule for how all life works, period.
And right here, this is where the plot absolutely thickens. Because scientists discovered a biological mechanism that just shatters this rule. A process that lets you take that temporary, flimsy RNA memo and write it back into the permanent, stable language of DNA. Now, this discovery didn't just bend the rules. It opened up an entirely new universe of what was scientifically possible. So let's talk about the core problem scientists were up against. That RNA message, it's incredibly fragile. It's meant to be temporary. But pretty much all of our most powerful tools for genetic analysis, especially for sequencing, are built to read the super-stable, double-stranded structure of DNA. So how do you possibly bridge that gap? This is the perfect way to visualize the solution. On one side, you've got your messenger RNA, or mRNA. Just think of it like that memo, written in disappearing ink. It's fragile and it's not going to last. But on the other side, you have what scientists can create. Complementary DNA, or cDNA. This is the archive. It's the permanent, library-bound copy that's sturdy, stable, and can be studied for years and years. The magic trick that makes all this possible is called cDNA synthesis. It's this amazing biochemical process that uses a very special enzyme called reverse transcriptase to literally read the RNA and write it backwards into DNA. This is the ingenious workaround that lets us turn a fleeting biological moment into a permanent record that we can actually study. Okay, so how is this archival copy actually made in a lab? It's not just a single magic step. It's a really carefully choreographed two-phase process. So let's pop the hood and see how it gets built, piece by piece. So it all happens in two main stages. First, in step 1, that special enzyme, reverse transcriptase, reads the original mRNA memo and builds one single strand of DNA that's a perfect mirror image of it. Then in step 2, a different enzyme comes along and uses that new DNA strand as a template to build the second, matching strand. And boom, you've got a complete, stable, double-stranded DNA molecule. Now, here's where it gets really interesting. While that first step is pretty standard, in step 2, scientists actually have a critical choice to make. See, there's not just one way to build that second DNA strand, and the method you pick has some pretty big consequences for the quality of your final copy. So let's look at the two main ways they do this. The older method, called self-priming, is, well, it's a bit clunky. It basically relies on the DNA folding back on itself, but as you can see, you can easily lose or scramble parts of the genetic code. But the more modern method, template switching, is so much more elegant. It uses an enzyme to gently chew up the original RNA, leaving behind these little fragments that act as perfect starting points. It is way more effective and avoids a lot of the harsh steps that could damage the final DNA. But of course, just like trying to bake the perfect cake, getting this right isn't just about following the steps. The success of the whole shebang really depends on carefully controlling a few key ingredients and conditions to make sure that final copy is flawless. There are really 4 pillars for getting a high-quality result. First, you have to start with a pristine RNA template. It's got to be clean. Second, you have to pick the right primer. That's the little hook that gets the whole process started. Third, the enzyme itself has to be top-notch. And finally, the reaction conditions, we're talking things like temperature, have to be absolutely perfect. And this chart just brilliantly shows one of the biggest challenges. The mRNA you actually want to copy, that valuable instruction memo, makes up this tiny, tiny fraction of all the RNA in a cell. We're talking just 1 to 5%. The other 95+% is just other stuff you don't want. It's like trying to find one specific important letter in a massive recycling bin that's overflowing with junk mail. So how do scientists fish out that 1 to 5%? Well, they use really specific fishing hooks called primers. For RNA from organisms like us, they use something called oligo-DT primers. These are designed to grab onto a unique little tail that's only found on those mRNA memos. For other things, like bacteria, or maybe samples that are a bit degraded, they use what are called random hexamers, which act more like a universal hook that can latch onto anything. And it's not just about finding the right RNA. The sample has to be incredibly pure. Any little leftovers from the extraction process, I'm talking about things like salts or ethanol, they act like smudges on the blueprint. They can literally stop that reverse transcriptase enzyme right in its tracks, preventing it from making a clean copy. So how do researchers even know if they're doing a good job? They measure something called the first-strand conversion rate. This number tells them what percentage of the original RNA was actually successfully copied into that first strand of DNA. And get this, a rate between just 12 and 50% is generally considered a success. That really shows you just how tricky this whole process can be.
All right, let's zoom out for a second and look at the huge picture here. Because creating these permanent DNA archives, it is so much more than just a neat lab trick. This single technique is the absolute foundation for some of the most powerful and revolutionary technologies in all of modern biology.
Seriously, every time you hear about RNA sequencing to understand a disease or profiling which genes are turned on or off in a cancer cell, that's powered by cDNA. When scientists produce life-saving proteins like insulin, they're often starting with cDNA. Even tracking how viruses like the flu or COVID-19 evolve relies on converting their RNA into cDNA first. It's everywhere.
And this really nails why it's so important. As the source material puts it so well, this technique is the crucial bridge. It connects the dynamic, constantly changing world of RNA inside a living cell to the powerful, stable world of DNA sequencing that lets us finally read and understand it.
When you boil it all down, that's what cDNA synthesis really gives us, a snapshot. It lets us hit the pause button to freeze a single moment in the life of a cell and capture all of its active instructions right then and there. We're taking a message that was about to disappear and turning it into a permanent piece of knowledge that can be studied forever.
It's an absolutely foundational tool that has already unlocked decades of incredible discovery. And it leaves us with a really exciting question. Now that we have this amazing ability to read life's most temporary and secret messages, what are we going to discover next?